Hi everyone, and welcome to our video on diversity in animal cells. This is part of our Module 2 Part 6 series looking at obviously all of those cell differentiation, cell division and cell diversity topics. In this video we'll be having a look at specification reference 2.1.6h, how cells of multicellular organisms are specialised for particular functions. First thing to remind ourselves of then is that when we talk about these specialised cells, these are ones that have become adapted to carry out a specific function in multicellular organisms. If we think about a single-celled animal, first of all, the amoeba, then we don't have any of these specialised cells because they're one cell. Now, the reason they can exist as this single-celled organism is because they've got this very large surface area to volume ratio. And that means that diffusion alone is going to be at a fast enough rate to meet all of the demands for oxygen intake and waste product removal. The way that the amoeba can actually divide the labour, because it doesn't have specialised cells to do particular things, is by actually using those different organelles to carry out a particular role. But there are not obviously specialised cells because there is only one cell. If we now think about multicellular organisms, they have a smaller surface area to volume ratio than those unicellular organisms. And what that means is that most of the cells that make up that organism are not in contact with the external environment. So what that means is that diffusion as a process is not going to be fast enough on its own to meet the demands of the organism. So in order to actually meet these demands, we require specialised cells to carry out specific functions within that multicellular organism. Now, the way that these cells are going to form is in the process of differentiation. So if we go to the very basic parts of the formation of an animal, we begin with obviously the process of fertilization. So we've got a spermatozoon which is going to be fertilized by an ovum, and we form a zygote. Now that zygote is a single cell with a diploid nucleus, but it's not specialized. Basically, this is a stem cell. It has a full set of DNA, all of the genes in the genome that could be expressed. Therefore, it could become any type of cell. What we then find is that original zygote is going to undergo several mitotic divisions, creating obviously a larger number of genetically identical cells. That's going to then form our embryo, and that is made of these embryonic stem cells. When we've got these embryonic stem cells, we're then going to see the process of differentiation take place. And differentiation is just where we are going to specialise that generic stem cell into a particular type of cell to carry out a specific function. And the way that this is achieved is by switching off certain genes within that genome. So what we end up with are different proportions in terms of the organelles present within those cells. We can change the shape of the cell, and we can even change the entire contents of the cell. And what we're going to do is have a look at a few examples of some of these specialised cells that you need to know about as per your specification. So our first specialised cell is the erythrocyte. You would have learnt this as the red blood cell back in GCSE. We're now going to upgrade our terminology to erythrocyte. The function of the erythrocytes is to transport oxygen around the body, and when we think about the actual erythrocyte itself, these are small cells with a large surface area to volume ratio, and that's going to be really important in that transport of oxygen function, because oxygen is going to be able to diffuse across the membranes of these erythrocytes easily, and that means it's going to let rapid diffusion take place. So we're able basically to load oxygen into these red blood cells, these erythrocytes, really quickly. We also have the kind of key shape, if you like, this very distinctive shape of the erythrocyte, a biconcave disc shape. And the reason it's got this biconcave disc shape is to increase that surface area to volume ratio. If we think about the specializations that are happening inside the cell, then there's no nucleus, mitochondria or endoplasmic reticulum, and actually very little cytoplasm. And the reason that we've basically done away with all of those different organelles is that we've got more space for hemoglobin. And hopefully we remember from earlier bits on proteins that hemoglobin is that protein molecule which is going to transport the oxygen. Second specialised cell we need to know about are the neutrophils. So the function of the neutrophils is to ingest bacteria and some fungi by phagocytosis. And these neutrophils make up around about 50% of all of those white blood cells within the body. If we think about the way that these are specialised, they're about twice the size of these erythrocytes. They contain what we term a multi-lobed nucleus. So that means the nucleus has got lots of little blobby bits sticking off of it when you look at the pictures. And the key thing about these is they're going to use a process called chemotaxis to actually move towards those sites of infection. So chemotaxis, if we break that word down, chemo is all to do with our chemicals and taxis is all to do with movement. So basically what we're seeing here is movement as the result of a chemical towards an infection site. Now, what we've got in the little bottom left corner here is a typical blood smear under a light microscope. We can tell it's a light microscope image because we can see many different cells present, and we can identify some of these key types of cell. So any of these ones that are these little kind of pinky ones with a white little center there, those are going to be our erythrocytes. What we then have are a range of these purpley coloured cells in here. Now, these are all different forms of white blood cell basically, and the one that we've been talking about so far is up in this top corner here, the neutrophil. So we talked about it having a lobed nucleus, so obviously we can pick out the lymphocytes themselves because they've got this purpley colour, and the dark purple in there is the nucleus. And do notice how we've got like these little lumpy bits. So you've got a lumpy bit on this end, got a lumpy bit on that end. That's the multi-lobed nucleus. Third type of cell we're going to look at is spermatozoa. So function is to provide that male genetic material during the process of fertilization. And the way that these have become adapted for that role is that they've got lots of mitochondria present within them, and they're located within this region here, the middle piece, where they're going to be carrying out lots of respiration, which generates ATP, and that ATP is required to actually move the undulopodium, that tail part of our spermatozoan. Now, obviously the movement of that undulopodium is all to do with allowing the cell to travel to the ovum for that process of fertilization to take place. Other bits we see here, they're small, long and thin. Obviously that means they can move through those different channels within the body nice and easily. And they've got this region at the front here called the acrosome. Now the acrosome is that region in the head of that spermatozoa which contains digestive enzymes. So once our little sperm cells actually reach the egg, what they've got to do is obviously get their nucleus into the same region as the egg's nucleus. So they've got to actually get through that membrane of the egg. So what happens is they're going to use the acrosome, and the enzymes within are going to digest the outer covering, which then allows the little sperm head to pop inside, and therefore fertilization has occurred. It is worth mentioning the fact that obviously the head is where we do have the nucleus, and remember in a human that will have 23 chromosomes present, for the simple fact it is half of the diploid number. It's that haploid number in our little gametes. We then have the epithelial cells. Now epithelial are like coverings. So their function is to form a covering on different surfaces in the organism, and there are two different types of epithelial cell you should be aware of. Squamous epithelial cells, and these are the ones which are quite flattened in shape, and these are going to make up things like the skin, for example. And then we've got ciliated epithelial cells. Now the ciliated epithelial cells are the ones that have got these little hair-like structures on the surface called cilia, and these are going to be found in regions like your airways, and their function in those locations is all to do with wafting mucus up the back of the throat so it can be swallowed down, and therefore anything trapped within the mucus is destroyed. As always, I hope you found this video useful, and don't forget to subscribe to the channel so you can see when the next video is uploaded, and of course head on over to the website to access a range of other resources designed to help you in your A-level biology studies.